This is a revision of Unit 1, and Unit 1 is Chapter 1, 2, 3, 4 that will be covered with the lecture exam 1. Chapter 1, as I mentioned before, is more of an introductory chapter. So if I'm going to ask you and what should you focus on in this part, it will be the homeostasis. Okay, so this is the most important part in chapter 1. Homeostasis, positive and negative feedback. Okay? What is a positive, what's a negative, and how does it go? So this is the most important part. Again, homeostasis, positive and negative feedback. For chapter 2, this is the chemical level of the organism or of the organization. In this part, you should differentiate between these terminology. This is actually important. What's the atomic number? What's the mass number? What is the element and the isotopes? What does isotopes means? And what's the atomic weight? These are the terminology that you need to know, each one of those. Okay? This is important to know what's the ionic bond, what's the covalent bond, what's the hydrogen bond, which one is the strongest, which one is in average, and which one is weak. And what does it mean, each one of those? Like sharing in covalent, ionic is cation and anion, which one donate and which one accept, and so on. So this part is important, and hydrogen is also important. What's the difference between molecule and compound? This is important. Okay? Different types of covalent bond. Covalent bond is important too. Single, double, triple. What does it mean? Hydrogen bond is important, and how do we make it? Which is the polar, related to the polar covalent. So hydrogen bond. So the three types of bond, very important. Three types. Okay, this is important. Decomposition versus synthesis, catabolism, anabolism, hydrolysis versus dehydration synthesis. You should understand the difference very well. Okay, what's the difference between hydrolysis versus dehydration synthesis? Which one is decomposition, catabolism? Which one is synthesis, anabolism? What's the function of the enzyme? Catalyst, lowering the activation energy for the reaction. This is important. What's the function of the enzyme? Okay. What's the difference between exergonic and endergonic reaction? This is also important. What do we mean by organic and inorganic? What are the three in biochemistry that tells you that this is organic? What are the two first? Practically speaking, what will be the third one? Oxygen. Okay. Nothing else. If you have those three, you're done. In the body. Outside, it's carbon, but I'm talking about biochemistry now. So this is what we need to know. If you want to say this is organic, carbon and hydrogen. Carbon and hydrogen in the body. Most likely, there is oxygen. But what is the minimum in the body? Carbon and hydrogen. Carbon is not enough. This is organic versus inorganic. Okay, hydrophilic versus hydrophobic, very important. You need to understand the difference between those two. And hydrophilic is also called lipo what? The opposite. Lipophobic and hydrophobic is called lipophilic. So anyway, it's the opposite of the lipids anyway. If you dissolve in the water, you don't dissolve in the lipids, basically. Okay, what's the difference between colloid and the suspension? These numbers are important. Neutral pH, the pH of the blood, you have to remember these numbers. And what's acidic, when you call it acidic, when you call it basic or alkaline, and how to get to that by increasing, the balance between hydrogen and hydroxide. Again, what's acid and what's base, and what weak acid versus and weak base means. Okay? What's salt? This is also important. Buffer. What's the buffer? This is important as well. For the carbohydrate, you need to know this ratio. So if I show you something, you need to count and tell me if it is carbohydrate or lipid or protein. The carbohydrate is just the ratio. You have to remember the examples of mono, di, and polysaccharides, including in the disaccharide, the third one. What is it? Lactose, yes. So the examples are important. And what's the minimum to say it's monosaccharide? 3 to 7. Can we say 2 or 8? No. Dehydration synthesis versus hydrolysis. We talked about this before and it's important. For the lipids, these are the different types of lipids. Just know what are these types. And understand the difference between saturated and unsaturated. And if it is unsaturated, is it mono or poly? Which is basically the double bond. Okay? And how to recognize that this is actually fat if you see COOH at one end. How about the proteins? Same thing, but the other end will have amino group, which is NH, NH2. Okay? So one side COOH and the other side is, can you recognize now the three? I think it's easy. Carbohydrate, 1, 2, 1. That's it. No COOH at the end. COOH at the end, we're talking about fat. COOH on one end and NH2 on the other end, this will be proteins. What's the function of leukotriene and prostaglandin? This is saturated or unsaturated? This fat. Unsaturated. Is it mono or polyunsaturated? Poly. Remember that you count only between the carbons. You don't count anything here. Okay, that doesn't count. What's a triglyceride? 1 and 3. 1 glycerol and 3 fatty acids. Head and tail, which one is hydrophobic and which one is hydrophilic? And what does it contain? Of the phospholipid, I mean. Protein. The main thing about the protein is it contains nitrogen. Carbohydrates, no nitrogen. Lipids, no nitrogen. What contain nitrogen? Proteins only. And in order to say this is a protein, not just a polypeptide, it has to be 20 or more. This is also important to know that all of these are proteins. Okay, buffers are proteins. Good part of them, not all of them. Enzymes are proteins. Hormones are proteins. Antibodies are proteins. Carrier are proteins, and so on. You need to know which of those is protein. What's a peptide bond? You need to understand that. You also need to know the shape primary, secondary, tertiary, and quaternary, just what each one of those mean and which one is the final shape. Characteristics of the enzyme, it's a catalyst, yes, we know, but it's specific, limited, and regulated. This is something that you need also to know. What's the difference between cofactor and coenzyme and isozymes? This is also important. What's denaturation? You lose shape and function by heat or pH. What's the difference between glycoproteins and proteoglycans? Which more and which less? This table is important, this one here, and this. Those two tables are important to differentiate between DNA and RNA regarding the sugar and regarding the bases. Okay? ATP, of course, is important. What's the function of ATP? What's the structure? And what's the enzyme that make or break ATP, which is ATPase. Okay, so this is for this chapter. That yes, I forgot that part. So how long the turnover, I forgot this. The turnover in the liver and in the muscles of the proteins and the glycogen. Specifically, liver and muscles. Neuron and fat, yes, that's fine. But liver and muscle, how long does it take for the glycogen to be depleted? Chapter 3. Chapter 3 is the most important of all. Okay? This is very important. What's the difference between sex cells and somatic cells? Specifically, the number of chromosomes, which is half in the sex cells. Okay, for the membrane, it is double layer of phospholipid, the arrangement of the head and tail, where is it located and why. Okay, and the function of each one of these proteins is also important. We have proteins within the membrane. Why? What's the function of integral protein versus peripheral proteins? Okay, these are the functions that we need to know.
So you want to know all the membrane proteins?
Proteins, yes, the function of each one of those. Why do we have it? What's the function of the glycocalyx? This is also important. Okay, the contents of the cytosol, very important. Very important. You have to know what is high inside versus outside and what's high outside versus inside. You have to know that. Specifically, proteins, potassium, and sodium. Okay? This is very important. The contents. Do we have proteins more inside of the cell or outside? For the proteins, it's inside the cytosol. What is the main extracellular cation and what's the main intracellular cation? Sodium and potassium and so on. So this is very important, the contents and the difference between the inside and outside. Okay, for this part, you have to know for each of the coming organelles. Number one, is it membranous or non-membranous? And then there will be like one function about each, or maybe two, about each one of those. And there is always a clue that can give you a hint to know the answer of the question. So we have microfilaments, intermediate filaments, microtubules. These are the different parts of the cytoskeleton. Which one is the largest? And which one is the intermediate? And which one is the small? Okay? Which is which?
Which one is the largest?
Tubules, microtubules. Microtubules, largest. Intermediate, the name gave it away. It's called intermediate already, so it's in between. And the microfilaments are the smallest. What's the function of the microtubules? It forms spindle apparatus. So for each organelle, you have to know the function. What's the function of the microvilli? Centriole, cilia. What's the function? The main thing: increased surface area, movement of fluid for the cilia. Centrioles, it's involved in mitosis. So each one of these functions. Ribosomes, what's the function? Protein, yeah. What's protein for what? Free or fixed? Proteosomes, what's the function? Again, this is the most important part. You need to tell me, is it membranous or non-membranous? And what is the main function? For the ribosomes, for example, protein synthesis. For what? For the cell or for export? Free or fixed? Proteosomes, what's the function? Contain proteases for recycling and so on. So membranous versus non-membranous. What's the function in the plasmic reticulum, and what's the difference between rough and smooth? Okay. So most important thing here is it make the three of them, and it also detoxify and store some molecules. But most importantly, what does these synthesize and what do and they detoxify? Smooth will make lipid and carbohydrates. Does it make proteins? No. Which one make protein? The rough. Golgi apparatus. What's the function? Okay. Packaging and modifying. Preparing it for export in the form of vesicle. What's the function of the lysosomes? And what's the difference between primary and secondary? This is important. And apoptosis or autolysis. Apoptosis or autolysis, which is a self-destruction by the lysozymes. When the enzymes are released, that's basically what it is. So lysozymes do apoptosis or self-destruction of the damaged cells. Yes?
So that's the same thing? Are you saying apoptosis and
Yes. And autolysis, it's the same thing. Apoptosis, 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 or autolysis. This is the same thing. Autolysis, self-destruction. When the lifespan is over, the cells will destroy itself. Using what? Lysosomes. What are the peroxisomes? Peroxide. They have peroxide. So break down fat and organic compounds. What's the function of the mitochondria? Most important thing. Energy. Powerhouse. Energy, powerhouse, which is ATP. What's the difference between aerobic versus anaerobic metabolism? Anaerobic is in the cytoplasm, produce only 2 ATPs. Aerobic is in the mitochondria and consists of glycolysis, TCA, electron transport chain. This is the difference between those two, aerobic versus anaerobic. Anaerobic in the cytoplasm, aerobic in the mitochondria. Anaerobic does not use oxygen, aerobic use oxygen. Anaerobic cytoplasm produce only 2 ATPs, aerobic will produce the rest, which is like 36. Nucleus, what's the function of the nucleus? Control center, control everything else. What's the function of the nucleoli? Not the nuclei, nucleoli. Okay, it make RNA histones, RNA and histones. What's the difference between chromatin and chromosomes? Okay, nucleosomes, chromatin, and chromosomes. What's the difference between DNA, gene, and genetic code? I mentioned it. This chapter is very important. What's transcription versus translation and processing? What are introns and exons? And how is that involved in the processing? How do you process? Which one do you remove, which is the introns, and which one you keep, exons? What's responsible for translation? For the codes, as I mentioned before, I only want you to know the stop and start. AUG, start. And the opposite, so AUG is stop. GUA, AUG, start. The opposite, stop. AUG versus GUA. AUG is, or ATG, it's the same thing, same code, because we replaced the T with U, if you remember, between the DNA and RNA. So ATG or AUG, start. TGA or UGA, sorry, let me write it down. AUG or ATG versus GUA, GTA. This is a start, and this is a stop. Can you remember this? That's it. Okay, what's permeability? What do you mean by impermeable versus free versus selective? And which one is a common one within our body, which is obviously the selective. What determine the permeability? Those four. Okay, in this part, which is passive and which is active is very important. Which one of those is active, which one of those is passive? This is the most important thing to remember. And in this part, just to summarize it and make it easier, only two things are active. If you're moving against concentration gradient or vesicular transport. Against concentration gradient or vesicular transport, meaning physical, which is you take part of the wall. Do you remember this? Those are the two active, anything else is passive. I'm just trying to summarize to make it easier for you. You don't have to memorize everything. If you know the active, anything else will be passive. What are the factors that determine the rate of diffusion? Distance, molecular size, temperature, gradient size, and electrical forces. This is something that you need to remember too. Osmosis is also important. The water moves from low to high or high to low. This is always confusing. I've seen that so many times. It always goes automatically in your mind. More to high, more to high. No, it's low to, it's low to high.
For the water.
For the water, we talk, osmosis is only for water. I'm not talking about diffusion. I'm talking about water only. Water only. Water move from less concentrated to more concentrated. The water move from dollar store grape juice to the good brand, which is more concentrated grape juice. Remember it the way you want. At the end, this is what it is. Osmosis, water move from less concentrated to more concentrated, from less osmotic to more osmotic. Remember it the way you want, just remember it. This is also important to know what's isotonic, hypotonic, and hypertonic. Same, less, more. And the example is here important, this one. What happened when you put an RBCs in isotonic? Stay. Hypotonic? Swallow, and then rupture, burst, and what do you call this process? Hemolysis. Hemolysis. Okay? Versus what happened if you put it in hypertonic? Which is called? Okay. What's the difference between co-transport and counter-transport? Co-transport together, counter-exchange. Okay, this pump is important to remember. Sodium potassium exchange pump. This is just one of the examples of the carrier mediated transport. And this is active. Okay? Why is it active? Because you're moving against concentration gradient. Remember, I told you the two active are against concentration gradient and vesicles. You don't necessarily, I don't have to ask you, you're moving against concentration gradient. Is it active or passive? That's too straightforward. It's not necessarily like that. If I ask you the sodium potassium exchange pump, is it active or passive? You need to ask yourself, are we using the sodium with the concentration gradient or against? Do you remember? Who's here yesterday or the day before? Are we moving the sodium against concentration gradient or with? Against. Is it active or passive? That's what you need to remember. So I don't have to clearly ask you, it is with the concentration gradient. No. If I told you sodium potassium exchange, ask yourself, are we moving sodium and potassium with concentration gradient or against? Against. So it has to be active. And 3 versus 2 for each ATP. Okay, so remember this. You exchange sodium, potassium against the concentration gradient, and for each through 3 sodium, you only move 2 potassium.
Do you have to know if they go in or out? Like any goes out, K goes in?
Yes. Which one moves out and which one moves in? Secondary active, secondary to something else, and just the example of that is glucose and sodium. You're moving sodium anyway, glucose will jump in. So sodium and glucose is an example of secondary active. What's endocytosis versus exocytosis? Are you entering something in or out? Is it active or passive? Yes. Why is it active? I told you already. The only thing that's active is against concentration gradient and vesicular transport. Isn't endocytosis and exocytosis is vesicular transport? Are you moving the vesicles out or in? Right? So it has, both of them are active. And panocytosis versus phagocytosis, what's the difference? Both of them are in, but what's the nature?
Fluid and then large objects.
The panocytosis is fluid, and the phagocytosis is nutrients or solids. Okay? So panocytosis versus phagocytosis. Both of them are endocytosis, and exocytosis is the opposite. What's the normal transmembrane potential? Is it negative or positive? It's negative inside compared to the outside. How negative? It depends on type of the cell. -10 to -100. Anything in between is okay. Cell life cycle. Just understand the steps. It goes like this. G1 or G0 to 1 to S to G2, and then mitosis and cytokinesis. Okay, this is one thing. The other thing is in mitosis versus meiosis, you end up producing same number of chromosomes or half number of chromosomes. And mitosis, do you end up producing same number? The daughter cell, does it contain the same number of chromosomes as the parent cell or half? How about the meiosis? It's the opposite. I'm sorry, but that's what it is. It's the opposite.
Mitosis is the same amount?
Yes. Meiosis. You know what's meiosis translation from Greek to English? Reduction. From 46 to 23. Reduction. This is just a translation. Meiosis means reduction. So you use meiosis to produce the reproductive cells, right? We know that the sperm and the ova contain 23, and all other cells contain 46. Do you remember that? So if you're making a liver cell, contain 46. You produce 2 liver cells. Each one will contain 46. For mitosis, 46. 146 will give you 246. 246 will give you 446. 446 will give you 1646. 32 and so on. 46, 46, 46, 46. You will never, ever make reduction of the number of chromosomes or it will be abnormal. All our body cells, everything except for the 6 cells, all of them contain 46 chromosomes. And when you reproduce those, you have liver cell that contain 46. You're making another liver cell. Why do you want it to contain 23 cells? Do you have like male and female liver cells that they will come together and make a baby or something? A baby liver? No, right? So liver cell contain 46. You're making another liver cell. Why do you want it 23? It has to be 46, right? You're making 46. 500. Each one contain 46. It doesn't change. The only time when you reduce 46 give you 23 is the 6 cells. And why? Because I have one from the male, one from the female. They will join together to make 46. So for meiosis, that's used in for the product of meiosis. If you're talking about the 6 cells, you start with 46, you produce 23. Anything else is mitosis 46. And by the way, mitosis means threading. You make a thread in between and retract to produce 2 cells. That's what it means. Meiosis, translation, it's Greek to English, reduction. What do you mean reduction? 46 to 23. Okay? Can you remember it this way? Just the translation. What's the name of the enzyme? Important. DNA polymerase versus RNA polymerase. You need to remember that. So if you're copying DNA to DNA, that will be DNA polymerase. DNA to RNA, which is called, what do you call this? DNA to DNA, what do you call it? The enzyme is called DNA polymerase, but what do you call it? The process is called replication. DNA to DNA, replication. And the enzyme is DNA polymerase. DNA to RNA, what do you call it? Start with T. Transcription. And what's enzyme? RNA polymerase, yes. Yes.
So you want us to know the steps of meiosis and mitosis, like anaphase and telophase and all that?
Just the general understanding. I don't need a lot of details. Just the general understanding. What does it mean? And which follow which. Okay, the MPF and growth factors versus the repressor genes is important. Which one accelerate and which one slow down? And which one, if interrupted or faulty, it can produce cancer for this part. Okay? What's the difference between benign and malignant? What's the most important thing? We're not discussing the details. Just what's the most important thing? Contained. If it's contained, it's benign. If it's not contained, means invasive, it's malignant. And it can also metastasize. So this is the most important thing to remember about this part. That's it for this chapter. For this chapter, which is 4, chapter 4, you need to know how to differentiate between the different types of epithelial cells based on the shape and the layers. So squamous, cuboidal, columnar, and is it simple or stratified? This is one thing. The other thing is if it is pseudo-stratified. What does it mean? Okay? And location. Specifically for this one, you have to remember the location. Okay? Location is important. Endocrine and exocrine glands. I think you were asking about this too? Okay. Yeah, endocrine and exocrine gland, this is also important. You need to know what's endocrine versus exocrine. Endocrine, you release hormones. Exocrine, you release enzymes. That's 1. 2, endocrine don't have ducts. Exocrine does have ducts. Okay? So this is the difference. Serous, mucus, and mixed, I talked about this. You need to know it's all about the secretion. Is it serous secretions, means watery? Is it mucus secretions, which is mucin, which is thick, viscous, a little bit thicker? Connective tissue, and I told you the rule without memorization. If it is not muscle, if it is not nervous system, if it is not covering or lining, everything else is connective tissue. The connective tissue consists of protein fibers and ground substance, and this will differentiate between the loose and dense. So what's the loose versus dense? What's the function of the connective tissue proper versus fluid versus supportive? What's the function of each one of those? And what's the difference between loose and dense? Again, it's all about the ratios. Fibers versus ground substance.
Can you repeat what you just said?
This one?
Right before that.
Yeah, what's the function of each one of those? Connective tissue proper, to connect, basically. Fluid, to transport. Supportive, to support, or strength. Okay? Loose and dense, ratios, which is more and which is less. This is very important. You have to know the function of each one of these cells, the simple function. Melanocytes produce melanin. Mast cells produce the two H's, and so as the basophils. Lymphocytes, immune. Macrophage and microphages, eaters. What make the macrophage? What make the micro? I actually can summarize everything in this slide right here. Okay? Fibroblasts make the substance and the fibers. Fibrocytes maintain. Macrophage and microphage, both of them are eaters. Macrophage come from the monocyte. Microphage will come from the neutrophils and eosinophils. Adipocytes, these are the fat cells. Mesenchymal cells, also known as stem cells, also known as the basal cells. Three names: mesenchymal, stem, basal. This is to reproduce and compensate or reproduce the injured or the dead cells. Melanocytes, melanin, mast cells, two H's, lymphocytes. So you got the functions already, and this is very important, each one of those. And the cement is called hyaluronate or hyaluronan. The difference between fixed and free macrophage, and where do they come from? Macrophage versus microphage, I just mentioned that. So the function of each one of the cells. The fibers, I want you to remember the location of each one of those. What's the difference between white and brown fat? If you know that brown fat absorb energy. White fat, on the other hand, is just a regular fat that we all know, which is everywhere for insulation, protection, storage of energy, storage of fat, and so on. Reticular tissue, location. Where is the reticular tissue? Liver, kidney, spleen, lymph node, and bone marrow. Location. What's the difference between tendon, ligament, and aponeurosis? Specifically, tendon and aponeurosis. What's the difference? Both of them do the same thing. Both of them attach the muscle to the bone. But this one is cord-like. Just mentioned that. Aponeurosis is sheet-like, flat-like. Perichondrium versus periosteum. And each one of those contain two layers. Which one is outside and which one is inside? You need to know this. Location is important. Elastic tissues, for example, is like the ligaments between the vertebrae. Fluid connective tissue for transport. This is examples. What is the example? Or what fluid connective tissue that we have? Plasma, part of the blood, lymph, and interstitial fluid, yes. So what's the lymph and what's the function of the lymph? It's still here. Okay. Lymph will take the extracellular fluid back, work as an immune system, and filter it out as it goes to the back to the heart. Perichondrium versus periosteum. I mentioned that. The outer is fibrous for strength and the inner is cellular for growth and maintenance. This is important to remember. Perichondrium and periosteum, both of them have the same thing. Outer layer, inner layer. Outer is fibrous for strengthening the tissue, and the inner is cellular for reproduction or growth and maintenance. So if you have injury, the inner one will replace your cells. For example, you have fissure or something in the bones, the inner one will replace it for you, not the outer. Location is important in the cartilage. Again, hyaline, stiff, and where is it located? Elastic, soft, where is it located? Fibrous, limit the movement, where is it located? This is what you need to know about this part. What make the bone? Collagen, fiber, and calcium salt. Important. Calcium salt is what make it hard. Calcium. Periosteum is the same as the perichondrium. Any membrane consists of epithelium and connective tissue. And examples here are important. Mucous membranes produce mucin, and what's the example? Serous produce serous, and what's the example? Cutaneous and synovial. Cutaneous is the skin, that's it. Synovial is around the joints, that's it. So you actually need to know examples specifically for the first two. Where is the mucus and where is the serous membranes? The main differences between those three. We will cover it in details later on. But at this point right now, what's the difference? Skeletal, voluntary, cardiac, and smooth, involuntary. Where is it? Skeletal attached to the skeletal system, which is the bone. Cardiac is in the heart. Everything else is smooth. The other thing is, the skeletal muscles are arranged in stria. So it is striated, parallel layers. While the cardiac is branching. While the smooth is random. It doesn't have a specific pattern. Okay, and of course, you don't have to memorize the location because it's everywhere, except attaching to the skeleton and in the heart. Anything else is smooth. Nervous system, what's the central and what's the peripheral? This is not details, we will cover that in so much details later. But brain, spinal cord is central and everything else is peripheral. Just a general idea. And what's the difference between neuron and neuroglia? Neuron is the actual nerve cell, neuroglia is just supportive cells. Just an idea about the structure of the nerve cell, not very important. Just know there's body, dendritis, axon, and telodendria, just generally speaking. And cardinal signs of inflammation, important. What are the cardinal signs that you can figure it out on your own? Just remember the last time that you had inflammation, and what did you have? And these three substances that are released in response for injury to elicit the inflammation process: prostaglandin, protein, and potassium ions. Regeneration, how does it work? Fibrocytes make collagen, and then epithelial cell come from the mesenchymal stem cells to regenerate. That's all what you need to know. For aging, everything deteriorates. If I ask you about anything, just deteriorate. Energy consumption, deteriorate. Metabolism, deteriorate. Hormones, deteriorate. Size, deteriorate. Decrease, lower, and so on. That's it.